Alrighty, guys, let's do it. So, let's talk about bowel cancer. ChatGPT, what's bowel cancer?
Bowel cancer, also known as colorectal cancer, is a type of cancer that affects the colon or rectum. It usually starts as benign polyps that can become cancerous over time.
Okay, let's focus on screening process and pathology too. So, okay, um, can you tell me some key facts about bowel cancer?
Sure, key facts. It's one of the most common cancers. Early detection through screening, like colonoscopies, improves outcomes. Risk factors include age, diet, and family history.
Why is it more common in Western?
It's more common in Western countries due to lifestyle factors like diet high in red or processed meats, lower fiber intake, and less physical activity.
Hmm.
Genetic predispositions also play a role.
So, food rich in red meat, why is this problem?
Red meat, especially processed, contains compounds that can form carcinogens during cooking. These can damage the colon lining over time, increasing cancer risk.
Whereas veggies are good, huh?
Exactly. Veggies are rich in fiber, vitamins, and antioxidants, which help protect the colon and reduce cancer risk.
So, what is long-standing ulcerative colitis and why is it a risk factor for bowel cancer?
Long-standing ulcerative colitis is a chronic inflammation of the colon. Over time, this inflammation can cause DNA damage in colon cells, increasing the risk of developing cancer.
Mm. And why is fruit and veggie helpful?
Fruits and veggies are high in fiber, which helps keep the digestive system healthy. They also contain antioxidants and anti-inflammatory compounds that protect cells from damage.
So, what does high-fiber diet do? Increases formation of short-chain fatty acids?
A high-fiber diet promotes the formation of short-chain fatty acids by gut bacteria. These acids help nourish colon cells, reduce inflammation, and protect against cancer.
And why is this good that they do this? It promotes healthy gut microorganisms? And also, why exactly do they increase the formation of short-chain fatty acids, the fiber? How do they do it?
Fiber acts as food for gut bacteria, which ferment it and produce short-chain fatty acids. These acids support a healthy gut lining and create an environment where beneficial microorganisms thrive, promoting overall gut health.
Okay, interesting. Guys, so if you haven't been to the channel before, we do multitask and jump from topics to topics. Hopefully I'll have put timestamps for you all. But yeah, today we're also going to talk about glycolysis and we're going to talk about the electron transport chain and mitochondria. So I'm so sorry we're talking about so many topics. At the end of the day, guys, this is for my revision too. So it's like, yeah, I know it's a bit too hard sometimes to figure it out. Okay. So let's jump a bit to glycolysis and we come back. So what's glycolysis?
Glycolysis is the process where glucose is broken down in cells to produce energy. It happens in the cytoplasm and generates ATP, the cell's energy currency.
Interesting. So
It's a
the
fundamental part.
timing of the processes, it doesn't really coincide with feeding times, so the storage forms of food are required.
Exactly. Since glycolysis provides quick energy, the body stores excess glucose as glycogen for later use when energy isn't immediately needed.
So what does ATP do?
ATP is the main energy carrier in cells. It powers various cellular processes from muscle contractions to chemical reactions by
Mhm.
releasing energy when its phosphate bonds are broken.
So ATP is in a complex with magnesium?
Yes, magnesium stabilizes ATP, helping it function properly.
Who
It's
knew?
essential for ATP.
Who knew? And so if I take magnesium supplements, does it actually help or is it more complicated than that?
It can help, especially if you're deficient. Magnesium supports energy production, muscle function, and more.
Why
But it's best
specific?
to check with.
Why specifically magnesium?
Magnesium is crucial for many biochemical reactions, including ATP production, nerve function, and muscle contraction. It's also important for bone health and maintaining a steady heartbeat.
What does it mean that magnesium is makes exchange susceptible to cleavage?
It means magnesium helps stabilize ATP, but in doing so, it also positions ATP's phosphate bonds in a way that makes them ready for breaking during energy transfer.
Hmm. What are bioenergetics?
Bioenergetics is the study of how energy flows through living systems, including how cells produce, store, and use energy, like in processes such as glycolysis and the electron transport chain.
What's anabolism and catabolism?
Anabolism is the process of building complex molecules from simpler ones using energy. Catabolism is the breakdown of complex molecules into simpler ones, releasing energy.
Okay, and let's introduce our last topic of the day, which is very related anyway, guys. Electron transport chain, mitochondria.
The electron transport chain is this super cool process in mitochondria. It's how our cells generate energy by passing electrons through a series of proteins, creating a gradient that ultimately powers ATP synthesis. It's like a microscopic power plant inside your cells.
And why is the electron transport chain also called respiratory chain?
It's called the respiratory chain because it involves oxygen. The chain's final step uses oxygen to accept electrons and form water, which is crucial for ATP production. This reliance on oxygen is why it's linked to respiration.
Interesting. Okay, so it's because it's related to oxygen. That makes sense. Okay, so it comprises four large multi-unit proteins intrinsic to the inner mitochondrial membrane. What is this? What is this?
Exactly. Those complexes are 1, 2, 3, and 4. Each complex plays a specific role in shuttling electrons and pumping protons to create that gradient. All these steps are crucial for ATP production.
And catalyzes a series. Okay, what is NADH?
NAD, or nicotinamide adenine dinucleotide, is an electron carrier. It shuttles
Wait.
electrons from.
No, I didn't say NAD. I said NADH.
Oh, my bad. NADH is a molecule that carries electrons to the electron transport chain, where it donates them to help generate ATP.
So what is this NADH plus hydrogen ion plus half oxygen equals NAD plus with water?
That equation represents how NADH is oxidized during the electron transport chain. NADH donates electrons, which combine with protons and oxygen to form water,
So
regenerating.
why does it need to get oxidized, and what is the NAD stand for?
NADH needs to get oxidized to release the electrons it carries, which are essential for ATP production. NAD stands for nicotinamide adenine dinucleotide. It's a coenzyme that shuttles electrons in metabolic reactions.
Hmm. And
Exactly. It's
why did we choose to have this metabolism be with reactions? Why did nature and evolution choose this?
nature's pretty efficient. Metabolic reactions evolved to extract energy from nutrients in a controlled, stepwise manner. This ensures maximum energy capture with minimal waste. And the electron transport chain is a great example of that efficiency.
Okay, and so energy release from this reaction, what does it have to do with ATP production?
The energy released from these reactions pumps protons across the mitochondrial membrane, creating a gradient. This gradient drives ATP synthase, which synthesizes ATP from ADP. It's
So
like water
you
behind a.
mentioned complex 1, 2, 3, 4. What is this? What is the differences? Sorry, I interrupted, but yeah, tell me.
No worries. Each complex has a specific role. Complex 1 accepts electrons from NADH, complex 2 from FADH2. Complex 3 and 4 pass these electrons down the chain, pumping protons to build that gradient for ATP production.
And what is ubiquinone, coenzyme Q? Is and cytochrome C? What do they have to do with all of this?
Ubiquinone, or coenzyme Q, shuttles electrons between complex 1 and 3. Cytochrome C then carries electrons from complex 3 to 4. They're like couriers in this energy relay race.
What do they do?
They transfer electrons between complexes, ensuring the flow continues smoothly. This flow is crucial for maintaining the proton gradient
And
that ultimately drives
they capture electrons.
Exactly. They capture and transport electrons, keeping the chain moving. This movement is what powers the proton pumps, building the gradient for ATP production.
Tell me more.
Sure, as electrons pass through the complexes, protons are pumped from the mitochondrial matrix to the intermembrane space. This creates an electrochemical gradient which drives ATP synthase to produce ATP.
Okay, so um, where do we find these names? Cytochrome C, ubiquinone, they're so specific, dude.
Ubiquinone and cytochrome C are crucial in the electron transport chain, which is part of cellular respiration. Their names are specific because they describe the molecule's roles and structures. Ubiquinone, for example, is a ubiquitous quinone found in many living organisms. Cytochrome C is a small heme protein associated with the inner mitochondrial membrane.
Jesus. Dude, I really want to spend time thinking critically about it. I'm running a bit behind on my stuff, but the next podcast would be more clear thinking. Okay, so we have flow of electrons and protons through complexes of the ETC, right? Right?
Exactly. Electrons move through the complexes of the electron transport chain in a series of redox reactions. This process helps pump protons across the mitochondrial membrane, creating a gradient that drives ATP synthesis.
So with factor 1, NADH becomes NAD plus, and then succinate becomes fumarate with factor 2, and then with the complex 2, sorry, complex 1. Complex 3, and what does complex 3 do in complex 4?
Yes, exactly. NADH donates electrons at complex 1, turning into NAD+. At complex 2, succinate is converted to fumarate. Complex 3 then passes the electrons from ubiquinol to cytochrome C. Finally, complex 4 transfers electrons to oxygen, forming water.
Hmm. Let's talk about the path of electrons entering the electron transport chain, please. So let's start from the beginning.
Sure, the electron transport chain starts with NADH and FADH2, which donate electrons. These electrons move through complexes 1 to 4, releasing energy that pumps protons across the membrane, creating a gradient. This gradient drives ATP synthesis, crucial for cellular energy.
What is FMN and FE-S?
Sure, when electrons enter the electron transport chain, they typically come from NADH and FADH2, which are products of earlier metabolic processes like glycolysis and the citric acid cycle. These electrons are transferred through a series of complexes embedded in the inner mitochondrial membrane, releasing energy that's used to pump protons across the membrane. This creates a proton gradient that drives ATP synthesis.
So what is FMN and FE-S? I know I've asked this again, sorry.
No worries. FMN stands for flavin mononucleotide, and FES refers to iron sulfur clusters. FMN is a prosthetic group that helps in transferring electrons within complex 1 of the electron transport chain. Iron sulfur clusters, on the other hand, are groups of iron and sulfur atoms that play a key role in electron transfer, helping to shuttle electrons between different proteins.
Okay. And this is all in the intermembrane space.
Mostly, no. The electron transport chain is embedded in the inner mitochondrial membrane. Complexes 1 through 4 are located there, and they create a proton gradient by pumping protons from the mitochondrial matrix into the intermembrane space. This
Okie
gradient is
dokie,
then used.
guys. Back to bowel cancer. Sorry for interrupting. Polyp. What's a polyp? Yeah.
No problem. A polyp is a small growth that can form on the lining of organs like the colon. Most polyps are benign, but some can develop into cancer over time. That's why doctors often remove them during colonoscopies. Catodoxis.
Interesting. So, what do we mean when we say most polyps in the large bowel are adenomas? What's an adenoma? What's the difference?
Sure, an adenoma is a type of polyp that forms from glandular tissue. Most polyps in the large bowel are adenomas, and while they're usually benign, they have the potential to become cancerous over time.
Can we
That's why they're
talk?
closely monitored.
Can we talk about low grade and high grade dysplasia, please? And yeah,
Absolutely. Low-grade dysplasia refers to mild abnormal cell changes in a polyp, indicating a minimal risk of progressing to cancer. High-grade dysplasia, on the other hand, means more significant abnormalities and a higher risk of developing into cancer. These are often removed to prevent progression.
what are the pathological features of polyps? What do we mean hyperplastic? Tell me again about this dysplasia.
Pathologically, polyps can vary. Hyperplastic polyps are typically benign and result from an overgrowth of normal cells, often with no cancer risk. Dysplasia in polyps refers to abnormal cells, with low grade indicating mild changes and high grade showing more severe abnormalities, signaling a higher cancer risk.
Okay, and can we talk about tubular adenoma and villus adenoma? What's all this?
Sure, tubular adenomas are the most common type of adenomatous polyps, typically small and with a lower risk of cancer. Villus adenomas are less common but tend to be larger and have a higher risk of becoming cancerous. They also have a distinctive finger-like appearance.
So we have hyperplastic polyp, tubular adenoma, tubulovillus adenoma, villus adenoma. Can you explain to me the differences again?
Of course, hyperplastic polyps are usually benign with no significant cancer risk.
Well, tell me the rest. I asked you about four things, sir.
Got it. Tubulovillus adenomas have features of both tubular and villus adenomas with an intermediate cancer risk. Villus adenomas are the least common, but have the highest cancer risk due to their size and structure.
So these are really important. What is the adenoma carcinoma sequence?
The adenoma carcinoma sequence describes how benign adenomas can gradually develop into cancer. It involves accumulating genetic mutations over time, leading to increasingly adenomal cells that can eventually become malignant.
What's the evidence?
The evidence comes from studies showing that genetic changes like mutations in genes such as APC, KRAS, and P53 accumulate as polyps progress from benign adenomas to malignant carcinomas. These mutations disrupt normal cell regulation, promoting uncontrolled growth and cancer development.
So, so can we talk about FAP? What is this FAP in bowel cancer?
Sure, FAP, or familial adenomatous polyposis, is an inherited condition where numerous polyps form in the colon and rectum. If left untreated, these polyps have a nearly 100% risk of turning into colorectal cancer. It's typically caused by mutations in the APC gene.
And why is it hereditary autosomal dominant?
It's hereditary because the mutation in the APC gene is passed down from one parent. It's autosomal dominant, meaning only one copy of the mutated gene is needed for the condition to appear. So, if a parent has FAP, there's a 50% chance they'll pass it on to their children.
All right, boys and girls, jumping to glycolysis. Can we talk about the structure of mitochondria?
The mitochondrion has an outer membrane and a highly folded inner membrane. The folds, called cristae, increase surface area for energy production. Inside the matrix contains enzymes for the Krebs cycle. The outer membrane is smooth, while the inner membrane's cristae are key to ATP synthesis.
What is this cristae?
Cristae are the folds of the inner mitochondrial membrane. They increase the surface area, allowing more space for the proteins involved in the electron transport chain, which is essential for ATP production.
So we have an inner membrane and an outer membrane.
Exactly. The outer membrane is smooth and encloses the mitochondrion, while the inner membrane is folded into cristae, creating a large surface area for biochemical reactions. This design is crucial for energy production.
Can we talk about cofactors?
Sure, cofactors are non-protein chemical compounds that assist enzymes in their function. They can be metal ions like magnesium or organic molecules like vitamins. Cofactors help in catalysis, stabilizing enzyme structure, or participating in the enzyme's reaction.
What are prosthetic groups?
Prosthetic groups are a type of cofactor that are tightly bound to an enzyme. Unlike other cofactors, they will remain permanently attached. Examples include heme in hemoglobin or flavin in flavoproteins. They play crucial roles in the enzyme's activity.
Mm-hmm. Interesting.
Glad you think so. Anything else you'd like to dive into?
Um, yeah. Can we talk about the just a sec. Mm, yeah, major redox coenzymes slash prosthetic groups involved in transduction of energy from foods to ATP.
Sure. The major redox coenzymes include NAD+, NADH, and FAD, FADH2. They carry electrons during metabolic reactions. ATP production involves these coenzymes transferring electrons to the electron transport chain in mitochondria.
So why do we have 2.5 ATP for NADH reoxidation? Why 1.5 molecules in FADH2 reoxidation? And why does oxidation give us ATP in general?
The different ATP yields come from where electrons enter the electron transport chain. NADH enters at complex 1, pumping more protons and generating about 2.5 ATP. FADH2 enters at complex 2, pumping fewer protons, thus yielding about 1.5 ATP.
Why does
Overall,
it
oxidation
why does it pump fewer protons?
FADH2 enters the electron transport chain at complex 2, bypassing complex 1. Complex 1 pumps protons, but complex 2 does not. FADH2's electrons result in fewer protons being pumped across the membrane, which means less ATP is produced.
Interesting. Let's talk about glycolysis a bit. So we have glucose and then we have glucose 6 phosphate, and for this to happen ATP becomes ADP because of priming reaction. You have to explain it to me. Then this is in the cytoplasm or something, and then we have fructose 6 phosphate from isomerase, then we have committed step where PFK1 it will become ATP to ADP and fructose 1,6 biphosphate. Can you explain all these steps to me? Huh?
Sure, glycolysis is basically the process of breaking down glucose to produce energy. It starts with glucose entering the cytoplasm, where it gets phosphorylated by hexokinase HK using ATP, which turns into ADP. This forms glucose 6-phosphate. Then, an isomerase changes it into fructose 6-phosphate. Phosphofructokinase 1, PFK1, then adds another phosphate group using ATP again, resulting in fructose 1,6 bisphosphate. These steps prepare glucose for splitting into 2 3-carbon molecules, which eventually leads to energy production.
I don't understand this PFK1, these isomerase. Why are these the catalysts? Why is hexokinase a good catalyst?
Great questions. These enzymes act as catalysts to speed up specific reactions in glycolysis. Hexokinase is a good catalyst because it efficiently adds a phosphate group to glucose, trapping it inside the cell. Phosphofructokinase 1 is crucial for regulating the pace of glycolysis, responding to the cell's energy needs.
And then,
Isomerase helps.
what about aldolase? Why does aldolase is good to make fructose 1,6-biphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate? Why specifically these? And why is isomerase making it this reaction that goes both ways?
Aldolase is essential because it cleaves fructose 1,6-bisphosphate into two 3-carbon molecules, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate. This step is crucial because it splits the 6-carbon molecule into units that can be further processed in glycolysis. The isomerase reaction is reversible, allowing DHAP to convert into G3P, ensuring both molecules can enter the next stage of glycolysis. This flexibility helps maintain efficient energy production.
And then this G3P, why is GAPDH the one that's going to make it 1,3-biphosphoglycerate? And why is NAD plus to NADH going to help this?
Glyceraldehyde 3-phosphate dehydrogenase is key because it catalyzes the conversion of G3P into 1,3-bisphosphoglycerate, 1,3-BPG. During this reaction, NAD+ is reduced to NADH, which helps capture the energy released from the reaction. This energy is used to attach an inorganic phosphate to G3P, forming 1,3-BPG, which then continues through glycolysis to produce ATP.
Why specifically GAPDH?
GAPDH is specifically suited for this reaction because it has a unique active site that binds G3P and facilitates the transfer of electrons to NAD+, forming NADH. This enzyme's structure and function are perfectly evolved to ensure the reaction proceeds efficiently, capturing energy in the form of NADH.
Why do I need to know this to become a doctor?
Understanding glycolysis is essential because it's a fundamental metabolic pathway. It helps you grasp how cells produce energy, which is crucial for understanding various physiological and pathological states. For instance, disruptions in glycolysis can lead to metabolic disorders.
Wait,
Plus,
so now ADP will become ATP. Instead, before we had ATP become an ADP. Why is PGK the one that's going to create 3-phosphoglycerate? And why is a mutase going to become 2-phosphoglycerate? And then enolase is going to become phosphoenolylpyruvate? Jesus Christ, dude.
I know, it can feel overwhelming. Phosphoglycerate kinase PGK uses the energy from 1,3-bisphosphoglycerate to convert ADP to ATP, producing 3-phosphoglycerate. Then, phosphoglycerate mutase shifts the phosphate group, forming 2-phosphoglycerate. Enolase then removes water.
Okay, so G3P, I don't understand why GAPDH is the best guy to turn it into 1,3-BPG, and then why that's the stage where NAD plus will become NADH. Can you explain that to me?
Sure, GAPDH is perfect for converting G3P to 1,3-BPG because it can bind G3P and facilitate the transfer of electrons. During this reaction, G3P is oxidized and NAD+ accepts the electrons, forming NADH. This also helps capture the energy released in a usable form. So it's crucial for energy production in cells.
Interesting. And then BGK will make it 3-phosphoglycerate and then mutase 2-phosphoglycerate. Why will this change? Why is this change significant? What are these SLP steps?
Exactly. PGK uses the high-energy phosphate from 1,3-BPG to produce ATP, creating 3-phosphoglycerate. The mutase then rearranges it to 2-phosphoglycerate. These steps are significant because they prepare the molecule for the final energy-releasing step,
And
called
then,
substrate.
and then enolase will make it phosphoenolpyruvate.
Right. Enolase removes a water molecule from 2-phosphoglycerate, forming phosphoenolpyruvate, PEP. PEP has a very high energy bond, making it perfect for the final step, where it donates its phosphate to ADP,
It's,
producing another ATP.
it's finally
And
gonna
that's how
become pyruvate.
exactly phosphoenolpyruvate donates its phosphate to ADP, creating ATP and forming pyruvate. This is the end product of glycolysis, and it can then enter further metabolic pathways.
And then each molecule of glucose gives 2 ATP, 2 pyruvate, and 2 NADH based on all these reactions we just saw.
That's right. Each glucose molecule results in a net gain of 2 ATP, 2 NADH, and 2 pyruvate molecules through these steps.
How can NADH be oxidized by the mitochondrial ETC, but the inner mitochondrial membrane is impermeable to NADH?
Great question. NADH can't cross the inner mitochondrial membrane directly. Instead, electrons from NADH are transferred into the mitochondria using shuttle systems, like the malate aspartate shuttle. These shuttles move the electrons, not the NADH itself, allowing the electrons to enter the electron transport chain for energy production.
So shuttles.
Exactly. They act like a bridge, moving the electrons from NADH across the mitochondrial membrane, so they can participate in the electron transport chain. This process helps ensure that the energy stored in NADH is efficiently used for ATP production.
So we have the glycerol 3-phosphate shuttle and the malate aspartate shuttle.
Yes, exactly. The glycerol 3-phosphate shuttle and the malate aspartate shuttle are the two main systems. They transfer electrons from cytosolic NADH into the mitochondria. The malate aspartate shuttle is more efficient, preserving more energy, while the glycerol 3-phosphate shuttle is simpler but slightly less efficient. Both ensure electrons reach the electron transport chain.
So the glycerol 3-phosphate and the malate aspartate, they're so specific. Why do we have these two?
They're specialized to suit different tissues and conditions. The malate aspartate shuttle, which is more efficient, is found in tissues with high energy demands, like the heart and liver. The glycerol 3-phosphate shuttle is simpler and found in tissues like the brain and skeletal muscle.
And why do they make
This
either 1.5 or 2.5 moles of ATP when they regenerate NAD+?
It depends on the shuttle used. The glycerol 3-phosphate shuttle yields about 1.5 ATP per NADH because it transfers electrons to FAD, forming FADH2, which enters the electron transport chain later. The malate aspartate shuttle is more efficient, yielding about 2.5 ATP per NADH because it transfers electrons to NAD+, allowing them to enter the chain earlier, generating more ATP.
Interesting. And what's DHAP?
DHAP, or dihydroxyacetone phosphate, is one of the two molecules formed when fructose 1,6-bisphosphate splits during glycolysis. It's converted into glyceraldehyde 3-phosphate, G3P, by an isomerase, so it can continue through glycolysis. This ensures all the carbon from glucose is used for energy production.
What is this? I don't understand.
No worries, DHAP is a 3-carbon molecule formed during glycolysis. It quickly converts into G3P, another molecule that can proceed through glycolysis. This conversion ensures all the carbon atoms from the original glucose are used for energy production.
Okay.
Got it. If there's anything else you'd like to dive into, I'm here to help.
And then inside we have FAD becoming FADH2 in another G3P dehydrogenase. What is this?
Exactly. In the glycerol 3-phosphate shuttle, electrons from NADH are transferred to FAD, forming FADH2. This happens via mitochondrial glycerol 3-phosphate dehydrogenase. The FADH2 then enters the electron transport chain, contributing to ATP production.
Mm-hmm.
It's a clever way to keep the energy flowing.
All right, guys, let's jump to the path of electrons entering the electron transfer chain. So let's talk about. Um, so we have FAD. Oh god, this is so complicated. I think I have to break it down. Let's talk about synthesis of ATP. We have ADP plus phosphate plus hydrogen, and then we have ADP, sorry, and then we have ATP and water.
No worries, we can definitely break it down step by step. So ATP synthesis happens during oxidative phosphorylation in the electron transport chain. As electrons move through the chain, protons are pumped across the inner mitochondrial membrane, creating a gradient. This gradient powers ATP synthase to convert ADP and inorganic phosphate into ATP and water. Feeling a bit clearer?
Mm. All right, guys, let's continue. Just so you know, there's been basically days worth of time lapse for me, guys. But yeah, let's pretend I'm continuing. Okay, no reason to anyway. I'm just joking. Genetics of FAP in bowel cancer, ChatGPT, enlighten us. Where is this APC gene? Interesting. What's the first hit? Interesting. And then they have the second hit. Hmm. And when and why and how does the second hit occur? Interesting. Okay. Um, let's talk about normal large bowel mucosa with no polyps. And yeah, dude, I can't lie. It looks disgusting. What are these yellow things on it? They like it's like extensions of it. And this is it in the intestine. Let's talk about early multiple adenomas in FAP. What is adenoma? No, no. Please, let's speak in English, dude. People don't understand Greek, yeah? Early multiple adenomas in FAP, and I want to understand what adenoma means. No, brother. No, brother. Adenoma. So it's like a volume thing. It's like, I see. And where is the bowel? And why are the two hits in the somatic cells? What other type of cells do you have anyway? Tell me this one more time. That makes sense. All right, a lot of questions there. So, can we talk about the two-hit hypothesis? Why is it called hypothesis? Mm-hmm. I see. Interesting. Okay, and can you explain to me this model? I can see here a diagram. Mm-hmm. Interesting. So, what is this thing? The second hit leads to loss of heterozygosity? Interesting. So, how does this happen? Okay. So, the first hit is basically from dominant dominant becomes dominant recessive. I see, I see. So, why can't we just be born with two recessive? Yeah. Let's talk about the malate aspartate shuttle, shall we? Why specifically malate and aspartate? This seems so specific, no? Interesting. Okay. And what do they do? So oxaloacetate is between them, and suddenly NADH becomes NAD. Where does the NADH come from? I don't understand. Does it catalyze the reaction or something? It basically gets oxidized, and that hydrogen is used for NADH. Yeah, but but before in the mitochondria, we have NADH becoming NAD, and I wonder why that happens. So, what's the point of going from aspartate to malate if only we're going to go back from malate to aspartate in a minute? Mm, I see. So, what's the role of pyruvate in metabolism? But here it says it can become lactate, alanine, acetyl CoA, or oxaloacetate, depending on if it's going to be lactate dehydrogenase, alanine aminotransferase, pyruvate carboxylase, or pyruvate dehydrogenase complex 2. And when does it become which? Hmm. Yeah, but I don't understand necessarily when it will become which. I understand which catalysts are going to be about, but go a bit deeper. So when sufficient oxygen is available, pyruvate can be oxidized to CO2 and water to generate ATP. And and so it binds to oxygen. Yeah, I don't understand how actually it generates ATP, the pyruvate. Why is it even called pyruvate, dude? Tell me more, please. I don't understand. Sorry, bro. I'm very confused. So we have pyruvate, which is a product of glycolysis. Okay. And then how does it go to ATP? Tell me step by step. What are these? What are these aerobic conditions? Anaerobic? I don't understand. Tell me again. So pyruvate with CoA and NAD will make acetyl CoA, CO2, NADH, and H. And why is this? Why do we need this? And why is the reaction irreversible? Mm-hmm. All right. Can we talk a bit about the TCA cycle, tricarboxylic acid cycle? Mm-hmm. Okay. And can you explain to me? I don't understand. So, TCA is also citric or Krebs. It's the same thing. Can we talk a bit about this? Why is it so important? So it occurs after glycolysis or before? So the products of glycolysis are used as the reagents in. Maybe I'm using the wrong word with reagents. Are used as the reactants in TCA? What is this acetyl residues? So we have four oxidation reduction reactions. One molecule of ATP is produced directly for each round of the cycle. So is there a reason that first we have NADH, then NADH, then FADH2, and then NADH? Why does it why does it show this efficiency? Why does this order maximize efficiency? How are they efficiently? Dude, you're speaking very generally. Can you talk to me specifics? You're like a politician now. Yeah, okay, exactly. But why is isocitrate and alpha ketoglutarate better off with NADH instead of FADH2? And don't tell me a general answer like efficiency. Tell me if the answer is efficiency, then why is it more efficient with NADH? Why? To be honest, dude, I still don't understand why this is the order. I'm still unsure. And the energy released from oxidations is conserved in the reduction of 3 NADH, 1 FADH2, 1 GTP, 2 CO2 also produced. How do we, why those so specific? Explain. Dude, I have to put hours and hours on this. I don't have the time right now, but I have to because it's like so specific. Talk to me about components of the TCA cycle that are important biosynthetic intermediates. Hmm, interesting. Okay, let's jump a bit into the ETC. So what's adenine nucleotide translocase? Guys, let me change the voice because it's not loud. Why is it known as anti-porter? So why does it do that? Why does it take ADP out? It's like squeezing the melon and then throw it away. Is it so? Actually, a mini phrase. Is it like squeezing a melon and then throwing it to the master that's going to fix it and renew it? Well, what did I say, sir? No, I said, what did I say? I said we're basically like using something, then recycling it. And who's giving them the ADP from the outside? Who's making it ATP? Is it the glycolysis or the Krebs? Hmm. Let's talk about let's go back to bowel cancer pathology. Let's talk about to progress for normal mucosa to adenoma to cancer genetic abnormalities. Let's talk about the steps a bit. I see. Okay, we talked about the APC. What's this antigen presenting cell? Interesting. Okay, and what is SMAD? Can we talk a bit about genetic abnormalities associated with bowel cancer? Lynch syndrome, FAP, we talked a bit about. Let's talk about attenuated FAP2, FCCX, MAP, serrated polyposis syndrome, Hamartomatous polyposis syndrome. Okay, let's talk a bit more about Lynch syndrome. Why is it called familial cancer? Familial. And why does it affect specifically the cecum and the right colon? Can you remind me where the cecum is? A cecum, is it in the ileum? Is it the edge of the ileum? Talk a bit louder, please. Mm. Let's talk a bit about the genetics of Lynch syndrome. So we have some pairs that can mismatch. Can we talk a bit more about that? Interesting. Can we talk a bit about MSH2, PMS1, PMS2? And sorry, before you continue, speak as loud as you can because I can't hear. Right. You know what? Maybe we should keep it for next time. Um, let's talk about uh, Amsterdam criteria. What's this? Mm-hmm. Let's talk about obstructing cancer. What is obstructing cancer? Huh? What? Interesting. Well, let's talk about sigmoidoscopy, colonoscopy. I assume sigmoidoscopy is basically examining the intestines. Mm-hmm. That makes sense. Yeah, let's do it. Interesting. Okay. Can we also talk a bit about CT colonography and staging CT scan for distal metastasis, please? Why is it CT better for metastasis? I don't understand the reason. Yeah, but I don't understand why MRI is not able to do that. Hmm. Alrighty. Can we talk about the staging of bowel cancer? T and B. I don't understand the difference between N and M. So T1 involves the invasion of the submucosa, and then T2 involves of the muscularis propria without full thickness invasion. Why? Hmm. And explain to me more T1, T2, T3, T4. Talk more about this. I'm gonna make a table. Tell me one fact for each one. Keep it 3 words for each fact. Well, okay. T4 invades other organs. T1 is muscularis. T2 and 3. Okay. And T3. T4. Mm. Oh, okay. What is this FIT stool test or fecal immunochemical test for bowel cancer screening? How does it work? Sir, let me change your voice and I told you how does it work? How does it work? Okay. So from blood. Okay. Let's go back to ETC and mitochondria. So, um, why do we need phosphate translocates? I don't know, actually, a better question is, why do we need phosphate and hydrogen ions into the matrix? Why do we need this importer? Okay. Can we talk a bit about ATP synthase? Can we talk about an F? What is F type? Can we okay? And what is this F0, F1 oligomycin sensitive proton channel ATP synthase? What is all this? Explain to me one more time, please. Dude, I'm so confused. Can you change the direction of how you're explaining this?
Sure. The FIT, or fecal immunochemical test, works by detecting hidden blood in your stool. It uses antibodies to specifically target human hemoglobin. So, you just collect a small stool sample at home, send it to a lab, and they check for any traces of blood. Easy and non invasive.
Oh yeah. Oh, sorry, guys. We already talked about that. Okay. Can you explain the science behind the stool test?
Sure. The FIT test relies on antibodies that bind to human hemoglobin. If there's blood in the stool, the antibodies will detect it. It's a specific reaction, meaning it only responds to human blood, reducing false positives from food or other substances. This makes the test accurate for detecting hidden blood that could indicate issues like colorectal cancer or polyps.
Hmm. Interesting.
Yeah, it's fascinating how such a simple test can provide
So what
crucial insights.
happens after a positive stool test?
If the FIT test is positive, it means blood was detected in the stool. The next step is usually a colonoscopy to find the source of the bleeding and determine if it's due to polyps, cancer, or another condition. It's essential for early detection and treatment.
Okay. Um, so why do the three, let's go back to mitochondria, guys, and ETC. Why do the three beta subunits have catalytic sites for ATP synthesis?
The three beta subunits of the ATP synthase enzyme have catalytic sites for ATP synthesis because they work together to convert ADP and inorganic phosphate into ATP. Each subunit undergoes conformational changes driven by the proton gradient, allowing them to bind ADP and phosphate, catalyze ATP formation, and then release ATP. This cooperative mechanism is essential for efficient ATP production in mitochondria.
Hmm. Um, all right. Can we talk more about these beta subunits and the gamma unit that rotates? I don't understand all of these things, dude. Let's talk about the structure. So we talked about the structure of ATP synthase, but okay. So there's alpha, beta, and gamma subunits. Okay, I can kind of picture it now. Let's talk about more about this.
Sure, ATP synthase has a complex structure. The alpha and beta subunits form a hexamer ring with 3 alpha and 3 beta subunits alternating. The beta subunits are catalytic, meaning they synthesize ATP. The gamma subunit acts as a central rotor, turning within the ring as protons flow through the enzyme. This rotation drives conformational changes in the beta subunits, enabling ATP synthesis.
Tell me one more time a bit more.
All right, picture ATP synthase like a molecular machine. The alpha and beta subunits form a ring, where the beta subunits are the workhorses making ATP. The gamma subunit sits in the middle, rotating as protons move through. This rotation causes the beta subunits to change shape, driving the production of ATP from ADP and phosphate. It's like a tiny spinning generator.
Electrochemical energy generated in intermembrane space, difference in charge and difference in proton concentration. What is all this?
Sure, the electrochemical energy comes from the proton gradient across the mitochondrial membrane. The intermembrane space has a high concentration of protons, creating a difference in charge and pH between it and the matrix. This difference, or proton motive force, drives protons back into the matrix through ATP synthase, powering ATP production.
Hmm, interesting.
It really is. It's amazing how cells harness energy so efficiently.
So, how much energy do we yield from one molecule of glucose and why?
From one molecule of glucose, we typically yield about 30 to 32 ATP molecules. This energy comes from glycolysis, the Krebs cycle, and oxidative phosphorylation. Most ATP is produced during oxidative phosphorylation, thanks to the proton gradient driving ATP synthase.
Wait, so we have 3 or 5 from glycolysis and ADH and then 280. I don't understand.
Sure, glycolysis nets 2 ATP and 2 NADH. Each NADH from glycolysis can yield about 1.5 to 2.5 ATP, depending on the shuttle system. The Krebs cycle produces 2 ATP, plus more NADH and FADH2, which contribute to ATP production in the electron transport chain. Overall, it totals around 30 to 32 ATP.
What are uncoupling reagents?
Uncoupling agents disrupt the proton gradient in mitochondria. They allow protons to flow back into the matrix without generating ATP, releasing energy as heat. An example is 2,4-dinitrophenol, DNP. These agents can be used by cells to generate heat. like in
So,
brown fat tissue.
so they put protons back to the matrix bypassing the ATP synthase. Why?
Exactly. They bypass ATP synthase, dissipating the proton gradient as heat. This can regulate body temperature in hibernating animals or newborns and can be exploited for weight loss. But it can also be dangerous if uncontrolled.
What do we mean it severs the link between electron flow and ATP synthesis?
Severing that link means the electron transport chain still moves electrons and pumps protons, but the energy isn't used to make ATP.
Then,
Instead, it's released.
sorry, instead what?
Instead, that energy is released as heat because protons bypass ATP synthase.
And what's DNP and UCP1? Brown adipose tissue, what's all this?
DNP, or 2,4-dinitrophenol, is a synthetic uncoupling agent. UCP1, or uncoupling protein 1, is a natural protein in brown adipose tissue.
So we have 5 to 10 copies of the genome in each mitochondrion. Why specifically 5 to 10? 900 different MT proteins are encoded by nDNA. What's all this?
Sure, the number of mitochondrial DNA copies varies because it depends on the cell's energy needs and mitochondrial turnover. More active cells have more copies to meet energy demands. As for the proteins, mitochondrial DNA encodes only a small portion of mitochondrial proteins. The majority are encoded by nuclear DNA and imported into mitochondria. This division of labor supports efficient mitochondrial function.
Explain to me again.
Absolutely. So, mitochondria have 5 to 10 copies of their DNA because it helps meet the energy needs of the cell. The number can vary based on the cell's activity. Most mitochondrial proteins are actually encoded by nuclear DNA, about 900 of them, and are imported into the mitochondria to support their function.
So most are imported into the mitochondria rather than made from them? I don't understand. Hmm?
No worries. Mitochondria have their own DNA, but it only codes for a small number of proteins. The majority, about 900, are encoded by the cell's nuclear DNA. These proteins are made in the cell's cytoplasm and then transported into the mitochondria to help it function properly.
Hmm. So what is this? So MT genome, it says codes for 13 respiratory chain, 13 of the respiratory chain proteins, 2 rRNA, 22 tRNA. What is rRNA?
The mitochondrial genome codes for 13 proteins that are part of the respiratory chain, which is essential for energy production. It also codes for 2 ribosomal RNAs, rRNAs, and 22 transfer RNAs, tRNAs. rRNAs are key components of ribosomes, the cellular machines that assemble proteins, while tRNAs help in translating genetic code into proteins.
Interesting.
You're welcome. If you have more questions, feel free to ask.
Um, Let me think.
Take your time. I'm here whenever you're ready.
So let's talk about maternal inheritance. The embryo essentially derives all the mitochondria from the egg. Most spermatochondria are in the tail, not absorbed on fertilization. Why?
Exactly. During fertilization, the sperm's tail, which contains most of its mitochondria, doesn't enter the egg. The mitochondria in the embryo come almost entirely from the egg, which is why mitochondrial DNA is inherited maternally. This inheritance pattern means mitochondrial DNA comes from the mother.
Why? Why from the mummy?
It's mainly because the egg cell is much larger and contains all the organelles, including mitochondria, needed for development. The sperm primarily contributes its DNA and loses its mitochondria, ensuring the mitochondria and their DNA come from the mother.
And what is this efficiency of OX oxidative phosphorylation, I assume? Declines with age. Why?
Oxidative phosphorylation efficiency declines with age due to accumulated damage to mitochondrial DNA and proteins. This damage can impair the electron transport chain, reducing ATP production and increasing reactive oxygen species.
That makes sense.
Glad it does.
All right, can we talk about more mitochondrial diseases and the heterogeneity of mitochondrial diseases? I don't even know what that means, dude.
Sure. The heterogeneity of mitochondrial diseases means they vary greatly in symptoms, severity, and affected organs. This is because mitochondria are involved in energy production for many different tissues. So depending on where the dysfunction occurs, symptoms can range from muscle weakness to neurological issues.
Tell me more. Tell me about what heterogeneity means.
Heterogeneity simply means diversity or variation. In the context of mitochondrial diseases, it refers to how these diseases can present differently in each person. This variation can be in symptoms, severity, or which organs are affected, making diagnosis and treatment challenging.
Tell me more about this. Please.
No problem. So heterogeneity means there's a lot of variety. In mitochondrial diseases, that means symptoms can be different from person to person. For example, one person might have muscle weakness, while another could have neurological problems. This variation makes it tricky to diagnose and treat because each case can be unique.
That makes sense. What is this homoplasmy and heteroplasmy threshold effect, genetic bottleneck?
Homoplasmy means all mitochondrial DNA copies in a cell are identical, while heteroplasmy means there's a mix of normal and mutated mitochondrial DNA. The threshold effect refers to the point at which the proportion of mutated DNA causes symptoms. The genetic bottleneck happens during egg cell development, where a small number of mitochondria are passed on, leading to variability in mitochondrial mutations in offspring.
Tell me again.
Sure. Homoplasmy is when all mitochondrial DNA in a cell is the same. Heteroplasmy means there's a mix of normal and mutated DNA. The threshold effect is the point where enough mutated DNA causes symptoms. The genetic bottleneck is a random selection of mitochondria passed to offspring, causing variability in inherited mutations.
I see. That makes a lot of sense. Okay, and let's talk more about the threshold effect now, please.
Sure. The threshold effect means that symptoms of mitochondrial disease appear only when the proportion of mutated mitochondrial DNA exceeds a certain level. Each tissue has a different threshold depending on its energy needs. If the proportion of mutated DNA is below this threshold, the cell can still function normally. But once it's crossed, cellular energy production is impaired, leading to symptoms.
Interesting. Okay, and then what is this mitochondrial genetic bottleneck?
The mitochondrial genetic bottleneck is a process during egg cell development. Only a small, random number of mitochondria are passed to each egg cell, which means the proportion of mutated to normal mitochondria can vary widely between eggs. This random sampling can lead to significant differences in mitochondrial DNA mutations passed to offspring.
Tell me more about this.
Sure. Because of this bottleneck, the proportion of mutated mitochondria can vary greatly between siblings, even with the same mother. This variability affects the severity and symptoms of mitochondrial diseases.
Let's talk about mitochondrial myopathies now, please.
Sure. Mitochondrial myopathies are diseases where mitochondrial dysfunction affects muscle cells, leading to weakness, exercise intolerance, and sometimes other symptoms. They're caused by mutations in mitochondrial or nuclear DNA.
So it reduces the capacity of cells to produce ATP. Can you talk to me about how some tissues like neurons, myocytes, skeletal muscle cells, beta cells of the pancreas are less? Okay, yeah, that's fair enough. Sorry, actually, forget that. I was just going to say that they won't tolerate this.
Exactly. Those tissues have high energy demands, so they're more affected by mitochondrial dysfunction. It can lead to significant symptoms in those areas.
Well, give me an example of mitochondrial myopathies then.
Sure. An example is MELAS syndrome. which stands for mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. It causes muscle weakness, headaches, seizures, and strokes, often starting in childhood.
Tell me more.
MELAS is caused by mutations in mitochondrial DNA affecting energy production. Symptoms can vary, but often include muscle pain, exercise intolerance, and neurological issues like strokes or seizures.
Okay,
There's no cure.
can we talk about LHON, MERRF, and MELAS and KSS?
Sure, LHON is Leber's hereditary optic neuropathy, causing vision loss. MERF stands for myoclonus epilepsy with ragged red fibers, with muscle twitches, seizures, and muscle weakness. MELAS we've covered. KSS, or Kern-Sayer syndrome, involves eye muscle weakness, heart issues, and muscle problems.
Let's talk a bit. Let's talk a bit about mitochondrial myopathies and the treatments and the prevention.
Are a group of neuromuscular diseases caused by genetic mutations affecting mitochondria, the energy powerhouses of cells. Symptoms can include muscle weakness, exercise intolerance, and fatigue. Treatments often focus on managing symptoms and might include supplements like coenzyme Q10, physical therapy, and dietary changes. Prevention isn't straightforward, since these are genetic conditions, but genetic counseling can help families understand their risks.
Interesting. So, what is this IVF strategy?
IVF stands for in vitro fertilization. It's a process where eggs are extracted from a woman's ovaries and fertilized with sperm in a lab. The resulting embryos are then transferred to the uterus. It's often used for couples struggling with infertility. This strategy can involve multiple steps, like hormone treatments, egg retrieval, fertilization, and embryo transfer.
And how come this works with mitochondria too?
In cases involving mitochondrial diseases, IVF can include a special technique called mitochondrial replacement therapy. This involves transferring healthy mitochondria from a donor egg into the mother's egg. By doing this, the embryo can have healthy mitochondria, potentially preventing the transmission of mitochondrial disorders.
Isn't this is the same as IVF?
It's similar to IVF, but with an added step. In mitochondrial replacement therapy, the nucleus from the mother's egg is transferred to a donor egg that has healthy mitochondria, but the donor egg's nucleus is removed. This creates an embryo with the mother's nuclear DNA and the donor's
I
healthy
don't understand.
mitochondrial
I don't
DNA.
understand.
No worries. So, in mitochondrial replacement therapy, the goal is to give an embryo healthy mitochondria. We do this by taking the nucleus from the mother's egg, which has her genetic material, and putting it into a donor egg that has healthy mitochondria. Then, this egg is fertilized with sperm. Does that help?
And could you explain to me one more time? And let's talk about this mitochondrial gene replacement from nuclear stage and then alternative strategy, maternal spindle transfer. What's this?
Sure. Let's break it down. In mitochondrial gene replacement, there are a couple of methods. One is called pronuclear transfer. This involves fertilizing both the mother's egg and the donor egg with sperm. Then the nuclear DNA from the donor embryo is replaced with the parent's nuclear DNA. Another method is maternal spindle transfer, where the mother's nuclear DNA is transferred to a donor egg before fertilization. Both aim to prevent mitochondrial diseases.
Tell me more.
All right, pronuclear transfer works after fertilization. The nuclear DNA from both the fertilized donor egg and the parent's fertilized egg are swapped. This ensures the embryo has the parent's nuclear DNA and the donor's healthy mitochondria. Maternal spindle transfer happens before fertilization. The mother's nuclear DNA is placed into a donor egg with healthy mitochondria, then it's fertilized. Both methods aim to prevent passing on mitochondrial mutations.
Interesting. So, so one is basically we get the egg out of the mommy and we do it, and the other one is what?
Exactly. One method involves fertilizing both eggs first and then swapping the nuclear DNA. The other, maternal spindle transfer, moves the mother's nuclear DNA into the donor egg before it's fertilized. Both aim to avoid mitochondrial mutations.
How is this possible? How can you do it before it's fertilized?
So, for maternal spindle transfer, the mother's egg is taken out, and its nucleus, holding her mitochondrial genetic information, is transferred into a donor egg that has had its own mitochondrial nucleus
All right,
removed. But
guys, we finished the episode prematurely. So well, not prematurely. We're finishing it properly. I'll see you at the next episode. Love you.